Ladies and gentlemen, please welcome the CEO of the Tom Price Foundation and the guests of honor of today's press conference. Distinguished guests, ladies and gentlemen, online viewers worldwide, welcome to the 2018 Tom Price Award announcements. From June 18th to the 21st, the Tom Price Foundation will be announcing the laureates of the 2018 Tom Price in Sustainable Development, Biopharmaceutical Science, Sinology, and Rule of Law. In today's press conference, the 2018 Tom Price laureate in Biopharmaceutical Science will be unveiled. To begin with, we would like to introduce the guests of honor who are here with us today. Please first welcome Dr. Zhang Wenchang, academician of Academia Sinica and president of Taipei Medical University. Dr. Yang Panchi, academician of Academia Sinica and professor of National Taiwan University. Dr. Gong Xingjian, academician of Academia Sinica and chair professor of Taipei Medical University. And Dr. Chen Zhenchuan, CEO of the Tom Price Foundation. Now we would like to invite Dr. Chen Zhenchuan, CEO of the Tom Price Foundation, to give us the opening remarks.
Distinguished guests, ladies and gentlemen, on behalf of the Tom Price Foundation and its founder, Dr. Samuel Yin, I welcome you to the announcement of the 2018 Tom Price Award for Biopharmaceutical Science. For the half-past century, biomedical science has been evolving at an astonishing pace. Much has been revealed regarding our bodies, leading to life-saving medicines, but much more has yet to be discovered. The Tom Price Foundation is honored to set up a world-renowned award in this field, rewarding scientists for their groundbreaking discoveries that have impacted human health. And by doing so, we hope the next generation will be inspired to follow in their footsteps. The foundation also extends special thanks to the hardworking, fair, and professional International Selection Committee. Congratulations to the Tom Price laureates to be announced, and wishing all of you the very best. Thank you.
Thank you very much. And now, ladies and gentlemen, we would like to invite academician of Academia Sinica, Dr. Zhang Wenchang, to announce the 2018 Tom Price laureate in Biopharmaceutical Science for us.
Distinguished guests, members of the media, I am now honored to announce the recipients of the 2018 Tom Prize in Biopharmaceutical Science. The recipients are Tanya Hunter, Brian Drucker, and John Madison. They are awarded for the discovery of protein tyrosine phosphorylation and tyrosine kinases as oncogenes, leading to successful targeted cancer therapies. Dr. Hunter received PhD from University of Cambridge, and now he is the Renato Dubecco Chair in Cancer Research Institute, Salk Institute, USA. Dr. Hunter discovered tyrosine phosphorylation and that the oncogene SAC is a tyrosine kinase. This discovery gave birth to the tyrosine kinase field and brought forth the golden era of cell signal transduction research. Dr. Brian Drucker received MD from UC San Diego and now is the director of Knight Cancer Institute, Oregon Health and Science University, USA. Dr. Drucker is a physician scientist who led the successful clinical trial imatinib, Gleevec, on chronic myelogenous leukemia, CML, which opened up the field of tyrosine kinase inhibitor, TKI, as target therapy. Dr. John Mendelsohn received MD from Harvard Medical School and now is the director of the Institute for Personalized Cancer Therapy, University of Texas, MD Anderson Cancer Center, USA. Dr. Mendelsohn demonstrated antibodies targeting epidermal growth factor receptor, EGFR, may be an effective strategy for cancer treatment, and got involved in the success of cetuximab, Erbitux, and its approval by the FDA to treat colon cancer and head and neck cancer. Thank you for your attention.
Thank you, Dr. Zhang. Next, please join me to welcome academician of Academia Sinica, Dr. Gongxing Jian, to introduce the Time Prize laureates in biopharmaceutical science and to give the award citation.
I'm really privileged to introduce or present to you the discoveries and the contributions by these three remarkable scientists. Dr. Tony Hunter is credited with the discovery of tyrosine phosphorylation reaction and the Src tyrosine kinase as an oncogene. Dr. Brian Drucker and Dr. John Mendelsohn established that tyrosine kinases can be superb therapeutic targets for cancer treatment. Dr. Drucker was studying BCR-ABL oncogene tyrosine kinase with the successful development of Gleevec to treat CML. Dr. Mendelsohn was studying EGF receptor tyrosine kinase with successful development of Erbitux to treat colon and head and neck cancer. With their work, we can now say cancers are treatable, and for some cancers, they actually have cures. Next slide. So before I introduce tyrosine kinases, I need to give a general background in probably the most oversimplified version about cancer, oncogenes, and tyrosine kinases. In its simple term, cancer is a disease of uncontrolled growth and spread caused by DNA mutations and altered gene expression. Among the 20,000 or human genes, there is a small set, perhaps 50 to 100, which upon mutations or overexpressions can lead to malignant transformation. They are called the drivers for cancer, and they are called oncogenes or cancer-inducing genes. In the past 30 years, there has been a lot of oncogenes discovered, and it is a garden variety with diverse enzyme activities, including protein kinase, G protein, transcriptional factor, and many of them are involved in transmitting growth signals. Among these oncogenes, tyrosine kinases, first discovered by Dr. Hunter, represent the largest family of oncogenes. They are the master switches of cell growth, survival, and excellent cancer biomarkers and therapeutic targets, as successfully demonstrated by Dr. Drucker and Mendelsohn. So what is tyrosine kinase. Tyrosine kinase is an enzyme that requires a cofactor called ATP, which provides its energy source and the phosphate. It can bind to ATP, but it's also other proteins. Its function is to place a phosphate onto the tyrosine residue, represented here as Y. And remarkably, when proteins with this tyrosine phosphorylate, it creates basically a link or a hook, if you will. It will attach to other proteins. And this protein can be tyrosine phosphorylate as well, and attach to another proteins. And so on and so forth. So with this protein-protein linkage, it can assemble a large number of proteins and activate them quickly. And this is how basically signal transduction is. And Dr. Hunter discovered this in 1980. And before that, it was known kinase can phosphorylate serine and threonine. But his discovery really was a breakthrough finding. Next slide. There are two types of tyrosine kinases, receptor tyrosine kinases and the non-receptor tyrosine kinases. Receptor tyrosine kinases are located on the cell surface, cell membrane. In fact, it cuts across cell membrane. Inside, their structure among different members are very similar because they all bind ATP and catalyze phosphate transfer to tyrosine. But outside, they're very different. And their structure is different because they bind to different growth factor. For example, EGF receptor that we'll be talking about binds EGF, and FGF receptor binds FGF. And there are also non-receptor tyrosine kinases present in the cytosol. And we will be talking about ABL a little bit today. And these kinases can also be active and transmitting growth signals. There's a strong connection between tyrosine kinases and oncogenes. Why? Because as we will discuss, they are the master switches of cell growth and survival. That's part of the story today. Next slide. So let's review briefly about how cells grow. The cells, when it starts to grow, it starts inside the nucleus with the DNA copied into two sets. And the cell mass also increased, double its cell mass, and become two sets, and then they divide from 1 to 2, 2 to 4, and that's how cell grows. But this process is tightly controlled. Only when if you have growth factor, there are soluble factors in the serum or between tissues, then when attached to the cell, will this process begin. And if we can enlarge the cells a little bit and look at how this works. Next slide. So what happened is we see this is that part of the cell, and we have a receptor situated on the cell membrane, and we have DNA in the nucleus. But there's no connection because the cells are in resting state. Next one. So when the growth factor binds, it will quickly transmit signals by activate other proteins, and some of them through phosphorylation. And some of the proteins are translocated into the nucleus, where it instructs the cell DNA to replicate, to transcribe, and to translate, to double the mass of the cell, including proteins and lipid. And the process of starting with growth factor outside and transmit the signal into the nucleus DNA is called signal transduction. Now, in normal cell, under normal conditions, this is, as I mentioned, tightly controlled. So let's remove this the growth factor. Then very quickly, everything disappeared. And in this case, the cell returned to its resting state. And this is how our body controls the cell growth. Now, what happened to cancer cell? The cancer cell, some of the key molecules, like the receptor and others, are mutated. When they mutated, they become constitutively active, and they can send the signals to the DNA to replicate without regard of growth factor. And in this way, the cell become growing and growing in an uncontrolled fashion. Now, it turns out, next slide, these two, for example, these two key molecules, many of them are tyrosine kinases. So tyrosine kinases are really the master switch of cell growth. Now, this is a cartoon drawing. In real world, next slide, you found actually, indeed, those color balls are true proteins with names. And this is one case that the receptor is a EGF receptor. Then when it mutates, it phosphorylates itself and linking the different proteins together. And in these two different pathways, it can activate, leading to growth and survival. Likewise, in some leukemic cells, we have a so-called ABL tyrosine kinase, BCR-ABL mutated. And ABL mutated, it also becomes constitutively active and sending the growth signal. So if we look at this, the increased cells, in cancer cells, these are the master switches that in order to stop its growth, the best way is to develop inhibitor to shut off this kinase activity. And that's exactly what scientists have been doing in the past 30 years or so. And so let's look at, there are generally two ways that you can inhibit tyrosine kinase. There are actually all other ways as well, but I would just use these two examples. So one way is that you can use small molecule that can diffuse into the cytoplasma, inside the cell. And they can bind to this mutated tyrosine kinase or kinase and blocking the binding of ATP to these molecules. Without ATP, the kinase will be inactive. The other way is to use antibody for cell surface receptor. It can block the binding of its natural ligand or natural EGF factor that can bind to here, and it can block the binding. That can also make the receptor kinase inactive. And this is exactly what the winners of this year's Tang Prize in Biopharmaceutical Science have done and elegantly demonstrated. So basically, Dr. Hunter discovered tyrosine kinase, a master switch for cell growth. Dr. Brian Drucker successfully developed a small molecule that can inhibit BCR-ABL tyrosine kinase. And Dr. John Mendelsohn utilized the antibody trick to block the EGF binding. Their work fully demonstrates or fully illustrates that the tyrosine kinases are the culprits of cancer, and they are druggable. Their success ushered in a new era for targeted therapy. So now I'd like to tell you a little bit about Dr. Drucker's work, very briefly. Dr. Drucker was and is interested in chronic myeloid leukemia, or CML, which accounts for 15% newly diagnosed leukemia, and is characterized by almost a universal BCR-ABL fusion protein. The BCR and ABL are proteins located on different chromosomes, but when the chromosome translocate, they fuse together. And once they fuse together, the original silent ABL now ABL becomes constitutively active. In so doing, it can transmit the signal. So in the mid 1980s, Dr. Drucker was at Dana-Farber. He began to be interested in the kinase inhibitor with Tom Roberts' laboratory. He continued the study at Oregon Health Science University with his industry partner, Dr. Nick Lydon. Together, with really clever strategy, they were able to identify small molecule now we know as imatinib or Gleevec structure shown in here, that when binds to the ABL, BCR-ABL gene, can block the ATP binding, thereby inactivate this kinase. And this is a really remarkable success, and it was the first FDA-approved tyrosine kinase inhibitor, and the first target therapy that successfully treated the majority of patients with CML. Even more impressive is the disease control rate is more than 90% with no recurrence in most patients undergoing maintenance treatment. These results are spectacular. Perhaps the next chart can even impress upon you even more. This is the impact of his work. That in 1982, before 1982, this is a CML survival rate. Only 8% patients survive. Whereas now, from 2001 to today, these are the number, 83% and 92%. And this is after 10 years. So many of these patients actually are cured of cancer. And this is a phenomena success. And this success, we really owe a great deal to Dr. Drucker and his team. Next slide. So now I'd like to tell you a little bit about John Mendelssohn's discovery. What he did was to use antibody to block EGF receptor. And why EGF receptor? As I mentioned, the EGF receptor is frequently overexpressed or mutated in different types, epithelial type of cancer. And Dr. Mendelssohn, when in the early 80s, was at UCSD, and he, together with his colleague, Gordon Sato, they conceived the idea, maybe an antibody can blocking the EGF receptor by preventing the EGF from binding it. That can be a good cancer treatment. And so together, and his other colleagues, they developed a monoclonal antibody originally called the C225. And now we know as Erbitux. So again, this is a tremendous success. It was the first FDA-approved antibody targeting EGF receptor for treatment of colon and head and neck cancer. I would just use the colon cancer as an example to show the impact of his approach. Now, colon cancer is a really dreadful, dreadful disease with very little prospect of treatment. And colon cancer results in close to 700,000 deaths each year worldwide, and is now the second most common cancer in Taiwan. So now let's look at an example of its impact. So this is for metastatic colon cancer. With the chemo alone, for these, the response rate is about 39% or 40%. But with Erbitux added, then it can increase to close to 60%. The median overall survival also increased significantly. But more importantly, this is a form of target therapy which can control cancer, relieve symptoms, and improve the quality of life. And so this is represents a remarkable process for a very difficult to treat disease. And Dr. Mendelssohn and his team deserve our true recognition. So in summary, the work of these three brilliant scientists has basically their work represents one of the most impactful discoveries in cancer research, which revolutionizes our understanding of oncogenic process and forever changes our outlook for cancer treatments. It also opens up two important fields in biology and medicine. The first is oncogene and signal transduction, and the second is target therapy. So both from the academic and the clinical perspectives, their work have huge, huge impacts. It is also a shining example of how innovative and the brilliant basic science could lead to successful biopharmaceutical and clinical applications with great benefits to society. And that is the true spirit of the Time Prize in biopharmaceutical science. Thank you very much.
Thank you very much, Dr. Gong. Thank you.